So, Assalamu alaikum. Assalamu alaikum, guys. My name is Rasoul Turko, and today I'll be teaching you about fatty acid oxidation and ketone body synthesis. Okay, so I know you guys have already learned something about fatty acids. You guys have learned about the synthesis of fatty acids. So when we talk about fatty acid oxidation, we're talking about the breakdown of fatty acids. So when do you think we'll need to break down fatty acids? Does anyone know? Let me look at the chat. No one has an idea? That's okay. All right, so we will break down fatty acids when the body needs energy, okay? When the body has been fasting for a while and it needs energy, because fatty acids are one of the major sources of energy that the body utilizes, okay? So for the learning outcomes, we're going to talk about mobilization of stored fatty acids. So those fatty acids that get stored, we're going to talk about how they move around the body. We're going to talk about the mechanisms of triglycerides degradation in the liver and the adipose tissue. So one thing to keep in mind is triglycerides and triacylglycerol are the same thing. Okay, they're a fat molecule that has a glycerol and 3 fatty acids. Hence the name triacylglycerol. So glycerol stands for the glycerol, and the acyl stands for the fatty acid. So triacylglycerol. Okay, glycerols. We're going to describe the oxidation of fatty acids, which is, again, the breakdown of fatty acids. And we're going to describe the ketone bodies and their use, ketogenesis, ketolysis, and ketoacidosis. So ketone bodies are what we get for when we break down fatty acids, and the result of that fatty acid doesn't get utilized in the ATC cycle, which I think you guys have learned about. Okay. So when we break down a fatty acid and we don't get, and we get acetyl CoA. So when we break down fatty acid, we get acetyl CoA. When we get that acetyl CoA, if we don't use it in the ATC cycle, then it turns into ketone bodies. Okay. And that gets me to my next point, which is that fatty acid. When we break it down to acetyl CoA, and we've learned. You guys learned in fatty acid synthesis how fatty acids are made from acetyl CoA. So this is basically the opposite. Okay. Okay. So so I'm going over Dr. Al-Jabbar's lecture. I decided to keep it as it is. I changed the order of some slides to help me explain what I felt like was needed to explain. So here he was talking about two different pathways. He was talking about exogenous fats and he was talking about the endogenous fats. So exogenous fats means when we eat something and we break it down. That's what it means by exogenous. So he goes over how it gets broken down in the stomach, and then and then with the help of the pancreas and their enzymes, it goes into the small intestine and then gets stored as triacylglycerol, which I said is the same as TAG or triglycerols. Okay, so it gets stored as that, and then it can get gets transported in the body through chylomicrons. So I'm going to go over chylomicrons and those things later, but for now, what you need to know is that when we eat something, it gets transported as a chylomicron. Okay. And endogenous fats, on the other hand, here he's basically talking about fats that are made in the liver. Okay. Second, yeah. Let's talk about the fats that are made in the liver. So those fats, when they're made in the liver, they go through the bloodstream. They get stored as triglycerides, but they are moved in the bloodstream through VLDLs or very low density lipoproteins. I'm going to explain this in my next slide, what they're talking about exactly in VLDLs. Okay, so again, and for those people that just joined, a triglyceride or a triacylglycerol is the basic fat storage in our body. When it gets broken down to a glycerol and 3 fatty acids. Now this glycerol can be used in gluconeogenesis or glycolysis. Okay, so basically, glycerol can give us glucose, which ultimately gives us energy, and that fatty acid gets broken down through beta oxidation, which we're going to be talking about right now, into acetyl-CoA, and that acetyl-CoA is essential in the TCA cycle. So you guys remember that whole circle with the different enzymes that yields some NADHs and FADHs that ultimately give us ATP. Yeah, so we need acetyl-CoA for that little cycle, and we get acetyl-CoA by breaking down those fatty acids. Okay, so again, fatty acids, and I need you guys to keep this in your head while we're going through this lecture, that fatty acids are the major fuel for the body during fasting states and exercise. Okay, so MOL2 is going to be MOL2 is basically like it's a whole course, and that has multiple lectures that you guys need to connect together in order to understand the bigger picture. And that's why there's some stuff that you need to keep in the back of your head when you're studying different lectures to better understand what they're talking. Okay, all right. So overview. So here's where it's really important. So like lipoproteins, there's going to be a whole 2 lectures on lipoproteins in MOL2, I believe. There's going to be plasma lipoprotein 1 and plasma lipoprotein 2, where they're going to go over this whole cycle here. But here, I think he put it to like further reiterate the process in which those fatty acids go through. Okay, so I'm going to go over it briefly. I don't think this is high yield for this lecture, especially. You guys will need to know it by the end of MOL2, which you will, inshallah, because there's going to be multiple lectures about it. Okay, so again, so when we eat something, it goes through, it's a dietary fat. It goes through the small intestine and ileum and all these things, and then it gets transported to chylomicron. You guys already learned about chylomicron, but a chylomicron, as you see those little blue, yellow, orange things. Okay, so the yellow things are going to be the triacylglycerol. Triacylglycerol, which I said are the same thing as the triglycerides, which are the same things as TAG. Okay. So it has a high density of triacylglycerol and a very low density of that blue thing, which is the phospholipid. Okay. That thing, that chylomicron, goes through the capillaries, and in the capillaries, it gets broken down by lipoprotein lipase. Okay, it's important to know that here the doctor is talking about breaking down the chylomicrons in the capillaries by lipoprotein lipase. It breaks it down into glycerol and fatty acid. Again, glycerol goes to be a precursor in glucose synthesis, whether it's gluconeogenesis or glycolysis, and the fatty acid gets beta oxidized to give us acetyl-CoA, which is that we then use. Okay, I'm gonna keep repeating this, and we're gonna keep going back to the same point, but it's hopefully like then it'll be easier for you guys to understand. Okay, after it goes to the hydrolysis in the capillaries, we get remnants of chylomicrons. So you see how those remnants have a lot less triglycerides because they got broken in the capillaries. Now that goes back to the liver, and in the liver, VLDL is made. Just a quick question: did you guys learn about LDLs so far?
We only got a little bit of information during this lecture.
Only this lecture, though, right?
Yes.
Yes.
Okay. So yeah, again, this is not going to be a very high yield for this lecture, but I'm just going to briefly explain it, but you guys will know it later, inshallah. So VLDL stands for very low density lipoprotein, okay, which means it has low density of the lipoprotein compared to the fat inside it. By fat I mean triglycerides. Okay, so those are made by the liver. They go through the capillaries, and through that we get IDL, which is intermediate density lipoprotein. And then after that, they go through the liver and they give us LDLs. Those LDLs is what we call bad lipoprotein. Okay, they're called bad because they have low density of the lipoprotein compared to the triglycerides. And you guys will know in later lectures why they're bad. And then after that, they get transformed into the peripheral tissue as cholesterol. And then from there we get an HDL. The HDL is high density lipoprotein, and that is the good lipoprotein, as people call it. I don't want you guys to get mixed up, so I don't want you guys to like pay a whole lot of attention on the slide, because yeah, again, you will learn a lot about it by the end of MOL 2. So like for now, let's keep focusing on the bigger picture. Is what happens with the triglyceride when it gets broken down. This is what I need you guys to focus on. Okay, so now let's zoom into this whole process here, which is going to be on the next lecture, on the next slide. Okay, so he's talking about the extracellular processing of triglycerides. Okay, again, triglycerides are the same as triacylglycerol. Why? Because there is a glycerol in the name. Let me write it. Triacylglycerol. So it's a glycerol. An acyl means fatty acid. Okay, and tri means 3. So that triglyceride gets broken down into a glycerol and 3 fatty acids. The glycerol again goes to as a precursor for glucose synthesis, and the acyl, which is the fatty acid, we'll be talking about here, and it goes through beta oxidation. Okay. So yeah, so those fatty acids get activated and transported into mitochondria. We're going to talk about this later, and they get broken down into acetyl-CoA, and that acetyl-CoA is essential to enter the TCA, the citric acid cycle, to give us the ATP we need, the energy we need. Okay, I'm just gonna look. Okay, sorry. Yeah. So okay. So now this is I want you. This is the capillary. Okay. So this is just a zoomed in version. So here's the endothelial cell. You guys know what the endothelial cell is, and you see those little like gray chains. Those will be the heparin, the polysaccharide chain, which is attached to the endothelial cell. And on that thing, on that little polysaccharide chain, we will have a heparin sulfate. Okay, the heparin sulfate. You guys talked about, I think, MOL 1. You guys talked briefly about those things, but you will need to know that heparin sulfate, which is on the endothelial cell, is gonna have the lipoprotein lipase. That lipoprotein lipase, which is right here, this like purple thing, attaches to something called ApoC2, apoprotein C2. That ApoC2 is basically just like a you can call it enzyme if you want. Like it's a little protein that is on the chylomicron. Okay, and that ApoC2 can be seen by that lipoprotein, and is basically telling the lipoprotein to come and break down that triglyceride. Okay, so again, ApoC2 is a protein that is on the chylomicron. The lipoprotein lipase is on the endothelial cell. It gets called by the ApoC2, so we can break down that triglyceride. Again, this whole thing is happening in the capillaries. Okay, we haven't talked about the stored fatty acids yet. We're talking about something that we eat. It gets into a chylomicron. That chylomicron goes into the capillaries. In the capillaries, it gets broken down. Okay, so this is a whole thing. This is just a kind of like this is something else than what the whole lecture is about. This is on the side. You guys just need to know that. Okay. Here I'm missing some letters, but it says allowing glycerol and the free fatty acid to be absorbed. Okay, this is what it says here. So yeah, so when activated by ApoC2, LPL, which is lipoprotein lipase, hydrolyzes or breaks down triglyceride in the chylomicron. Okay, all right. So now that was an extracellular pathway. Now we're talking about intracellular pathway. So I found this this little picture that really gets to the point. So you see the person right here. So this person right here is symbolizing like a person in a fasting state. So I know someone was asking me in the chat about fasting state question mark. Does anyone have a question about that? Okay, I'll just explain what I mean. What I mean by fasting state, I mean, it's someone that hasn't eaten something in like someone that hasn't, yeah, someone that's not eating something right now. That's the person that's in a fasting state, because this is really important. We talk about when we're talking in MOL, because we have some enzymes that get produced when someone eats something, which you guys talked about, like insulin and those things. And there's other enzymes that get produced when someone is in a fasting state, or someone has, yeah, and someone's not eating. Yeah. Okay. So a person is in a fasting state. That means their blood they don't have their blood glucose is down. When their blood glucose is down, that means their energy is all they have less ATP as well, because they have low glucose. When we have low blood glucose, it signals to the pancreas to produce glucagon. Okay, glucagon. I think you guys learned about glucagon, but it basically increases blood glucose in our body through different mechanisms, through gluconeogenesis and multiple different things. So when it produces glucagon, this glucagon activates something called an enzyme called hormone sensitive lipase. Okay, so back in the first slide, in the slide before it, we were just talking about lipoprotein lipase, which is in the capillaries. Here we're talking about someone like myself that has like a fat cell in their body. They're in a fasting state. They haven't eaten in a while. We produce glucagon. That triglyceride, that fat cell that's in my body, that's stored in the adipocytes, the adipose tissue, gets broken down by hormone sensitive lipase. It gets broken down. It's the same process as lipoprotein lipase. The same process as lipoprotein lipase, because it breaks down the triglyceride into a glycerol and 3 fatty acids. Okay, but here we're talking about it in the sense of it being as already stored fatty acid. Up there, we're talking about something that has just a chylomicron that was just ate. Okay, so it activates hormone sensitive lipase, breaks it down into glycerol and 3 fatty acids. Now this fatty acid, this is what a fatty acid is, by the way. Sorry. Yeah, those are 3 fatty acids. Okay, that fatty acid is lipophilic, right? So that means it's not soluble in blood, which means it needs to form a bond with something that makes it easier for it to be transported around the body, and that's where albumin comes in. So albumin will attach to that fatty acid, and then it can get into the capillary it can get into the circulation and get to the target cell, which is the liver, for example. So we can then begin our beta oxidation and break down the fatty acid. Make sense? Okay. By the way, for the people that just joined, if you guys have any questions, stop me at any time. Okay. All right. So I'm gonna skip this slide. I'm gonna go over this slide first before going over this. Okay. All right. So again, now what we've gone through is breaking down that triglyceride into 3 fatty acids and a glycerol. Okay, and the person is in a fasting state, and they have low blood glucose, low blood glucose, which means low ATP, which means we need an enzyme, which glucagon is the enzyme that we need. So that glucagon, now we're talking on a cellular level here. Glucagon comes and it activates something called adenyl cyclase. Okay, so this is the receptor. So here we're in a cytoplasm. Here, this is the cytoplasm. This is the bloodstream. Here, in a cytoplasm, there's a receptor for glucagon. Glucagon binds to that receptor. That receptor activates adenyl cyclase. Adenyl cyclase then uses the ATP to give us cyclic AMP. Cyclic AMP is an intracellular it's an intracellular it's an extracellular signal, which then activates pKa. So the way I like to memorize this, and it's just an easier way, because you guys are gonna go over this pathway a lot in MOL. In MOL, you guys might go over it in heme, I believe. You guys will go over this exact like first 3 steps a lot. So adenyl cyclase, there's an A, and adenyl cyclase. So A, there's a cyclic AMP, okay, A again, and there's a pKa, A again, okay. So adenyl cyclase activates with the help of ATP, gives us cyclic AMP, and that gives us pKa, okay. Now, pKa, which is protein kinase A. Kinase, when we say kinase, it means it will phosphorylate something. By phosphorylate, we mean it gives it adds a phosphate group to that something. Okay, so protein kinase A does 2 things. Okay, it phosphorylates or adds a phosphate group to HSL, which we just talked about is the hormone sensitive lipase, and it phosphorylates something called perilipin. Okay, when it phosphorylates that, so perilipin is right here. It phosphorylates it. It adds the phosphate to it. When it adds the phosphate to it, it gets it gets attached to CGI, which is right here. Okay, so by phosphorylating perilipin, we can get it to bind to CGI. Okay, now the binding of CGI. Sorry, when we phosphorylate perilipin, we let CGI go. Okay, it doesn't bind to CGI, it lets it go. When CGI is by itself, then it can bind to ATGL. ATGL is what? Adipose triglyceride lipase. Okay, which means adipose, fat cell, triglyceride, again, TAG, lipase, breakdown. So it's the first step that we need to break down triglyceride, okay? ATGL. Okay, after that, when we break down triacylglycerol or triglyceride, same thing, we get diacylglycerol. That diacylglycerol then gets broken down by the phosphorylated HSL, this HSL that has already been phosphorylated by pKa. We needed to break down diacylglycerol to something called monoacylglycerol. After we get monoacylglycerol, with the help of MGL, we will get, what's it called? 3 fatty acids. Right here, get 3 fatty acids. Yeah. And then when we get those 3 fatty acids, so by the way, we're still talking about the same process that we talked about in the last 2 slides. Okay, this is just more on the cellular level. Okay, so we said that glucagon activates that whole cycle, but so that means that insulin will do the opposite. So when do we get insulin? We get insulin when we have high blood glucose or high ATP in the blood. So that means when we have high ATP, that means there is no need for us to break down the fatty acids that we have. And the triglyceride, that's the fat cell that's in my body. There's no need to break it down if I'm eating and I have enough blood glucose, right? So when I have enough blood glucose, that means I have there's insulin. When we have insulin, it activates a phosphatase. So phosphatase is the opposite of a kinase. A kinase adds a phosphate group. A phosphatase takes that phosphate group out. So when that phosphatase gets activated, it takes the dephosphorylase HSL, which will inhibit basically this step of breakdown, and that promotes fat synthesis because we're not breaking down the fatty acids. That means it promotes fat synthesis. Okay. Now, after going through that, I'm gonna make you. You guys can look at this slide.
I have a question.
Yes.
Can you repeat what will do protein kinase A? This particular
protein kinase A. Okay, so protein kinase A, a kinase phosphorylates usually. So it's adds a phosphate group. Okay, so protein kinase A does 2 things in this step. It phosphorylates HSL, hormone sensitive lipase. That means it adds a phosphate group. So here it didn't have a phosphate group. and here it has a phosphate group, okay? And it phosphorylates perilipin. This perilipin, this little like gray, this purple thing, it phosphorylates, it adds a phosphate group to it, like we see here. Okay, so it phosphorylates two things. And that's what a kinase means. A kinase just means that it gives a phosphate group. Okay, makes sense?
Okay, thank you so much. Okay.
Of course. Okay. Yeah, so if you guys, we're gonna go back to the same slide. So this slide is the exact same thing that's happening right here. TG, as in triglyceride, with the help of ATGL, adipose triglyceride lipase gives you diglycerol, and then hormone-sensitive lipase gives you monoglycerol, and then MGL breaks it down to glycerol, glycerol, and 3 fatty acids. Okay, those are 3 fatty acids. Okay. All right. And again, we talked about how those fatty acids, in order for them to move around the body, they need to bind to albumin, which is a protein that it's a transport protein. Okay. All right. So we've reached our first checkpoint. Does anyone have any questions so far? Okay, then I'll keep going. All right. Okay, so we talked about glycerol. Okay, so here the doctor just put that he wants to just show you guys that glycerol is needed for glucose synthesis. And here, this is what it's basically saying. But I found this picture that shows you. You guys probably remember those guys, all those enzymes. So this is like glycolysis. This is glycolysis. This whole thing. So we see how glycerol, after we break it down, after we break down triglycerides, we get the glycerol. Now that glycerol, through different enzymes, those enzymes, we get dihydroxy acetone phosphate, which then can be used into breaking down of glucose in glycolysis to get energy. So you see how after breakdown of triglyceride, we can use that glycerol. We can use that glycerol even to make energy. Okay. Those enzymes right here, I don't believe we're very high yield in our exam. The whole point is what happens. Those enzymes involved in beta oxidation. Okay. You guys just need to know that glycerol is a precursor, or can be a precursor in glycolysis or gluconeogenesis. Okay, that's all you need to. That's all you guys need to know. And honestly, from the name, you can tell what they're talking about. Like glycerol kinase, it basically as a phosphate group. So it's from glycerol, it adds a 3-phosphate. Again, phosphate dehydrogenase, same thing. Glycerol phosphate dehydrogenase. So you can tell from the name what they're trying to tell you. Okay. Okay. Now, step 2. So now we have broken that TAG, triglyceride, into fatty acids. Now, what are we gonna do with those fatty acids? Those fatty acids are right now in the cytosol or cytoplasm. Okay, so they will need to be activated, and then they will need to be transported inside the mitochondria. And in the mitochondria, they go through 4 different reactions, and to give us the acetyl CoA we need, and NADH and FADH. Okay. So we have a fatty acid. So this is just an overview of the whole process. I'm just going to go over it real quick, and I'm going to go in detail in the next couple of slides. So now we just have a fatty acid. Then it gets activated. This is the name of the step here. It gets activation. So it gets activated by this CoA. Okay. So it's from a fatty acid we get a fatty acyl CoA. And it's important here, fatty acyl CoA. And this is an important step. Why? Because here we use in this process of activation, like this here, in this process of activation here, we use 2 ATPs. Okay. So basically we're using some ATP to get a whole lot of ATP. So I think a C16, a palmitoyl C16 fatty acid gives us around 190 something ATPs. So we're using only 2 to get all the ATP. Okay, so this is important because in this step, in the activation step, and we're going to talk about the enzyme that is used here. But in the activation step, we use 2 ATPs. Okay, after that we have a fatty acyl CoA. That fatty acyl CoA binds to carnitine. Binds to carnitine, then gets absorbed. Not absorbed, gets transported inside the mitochondrial matrix, and then in the mitochondrial matrix, we get a fatty acyl CoA again, and then it goes through dehydrogenation, hydration, dehydrogenation, and then catalytic cleavage. So this is the overview of the whole process. It's going to be divided into the next couple of slides, but it's easier to look at it from like a bigger picture because it's basically 3 steps. It's the step of activation. It's a step of transporting it from the cytoplasm inside the mitochondria, which we're going to talk about. And then it's last step is basically just 4 like small steps. Okay. All right. Okay. So fatty acid activation. So fatty acid activation is what we talked about over here. The whole thing is the fatty acid activation. Okay. So the fatty acid activation is ATP dependent. Okay. Which means we use ATP. We need ATP in order for us to activate the fatty acid. So that fatty acid, this is the fatty acid here, it becomes a fatty acyl CoA here. So this is the fatty acid here, and this is the CoA. So it becomes a fatty acyl CoA. And here, notice how it goes from ATP to AMP. So I know it's only one process, but because it's going down from ATP straight to AMP, and it's skipping ADP, and that means it's going from ATP, ADP, and then AMP right away. So skipping one, that means it's using 2 ATPs. So it's using 2 ATPs for activation, and the process of that pyrophosphate, which it's called hydrolysis of pyrophosphate, where the pyrophosphate is the ATP, and we just hydrolyze it to get AMP. Because it's going from ATP to AMP, it makes this whole step irreversible. Okay, this is what you guys need to know. And this whole activation is by this enzyme called long chain fatty acyl CoA synthetase, or LCAS in short. Okay, that whole process of activation, which again is from fatty acid to fatty acyl CoA, that is given by the long chain fatty acyl CoA synthetase. And I really want to stress on the fact that like I need you guys to like read the name and try to understand what they're talking about, because it's going to make your life a lot easier to memorize all those names. So here they're saying long chain fatty acyl, we talked about fatty acyl is acid, CoA player synthetase. That means it's a long chain fatty acid and fatty acid with the CoA, and like with right here. So fatty acyl CoA synthesis. So it makes that thing. So it's basically the enzyme is telling you what it does basically. Okay. Here the doctor put a little side note on a disease that can happen. So when someone has a deficiency in LCAS, that means that fatty acid cannot be activated to get into beta oxidation. That means we have a lot of long chain fatty acids in the brain and adrenal cortex, and that usually gives us adrenoleukodystrophy. Okay, ALD. That's all you guys need to know. Yeah, he didn't mention anything else. Okay, that's the first step. Now, second step. So now that we have fatty acyl CoA, okay, it got activated by LCAS. Now we have fatty acyl CoA. That fatty acyl CoA needs to get inside the mitochondrial matrix from the cytosol. Okay, and it needs an enzyme to do. It needs 3 enzymes actually to do that. Okay, first of all, we get the fatty acyl CoA. After that, it binds to carnitine. It binds to carnitine because the carnitine would allow it to go in. Without the carnitine, they cannot go in. A fatty acyl-CoA cannot go in. So it is carnitine. That is given by carnitine palmitoyl transferase 1, which is CPT1. So the process of adding a carnitine to the fatty acyl-CoA is by that enzyme. Okay. Now that we have an acyl-carnitine, that acyl-carnitine can slowly go through the extramembrane to the inner membrane. So now it's in the inner membrane here. After that, we need an enzyme called translocase to bring it from there from the inner membrane inside the matrix. Right when it gets inside the matrix, that's when the job of carnitine is done. We don't need carnitine anymore, and that's when CPT2 or carnitine palmitoyl transferase 2 comes and takes that carnitine out. Now we have fatty acyl again. So we basically the same thing that we have outside, we will have it inside. So it was an acyl-CoA outside. It got transformed to acyl-carnitine. The acyl-carnitine got in by CPT1, and then the acyl-carnitine got inside the matrix by translocase. Now the acyl-carnitine through CPT2, we will get acyl-CoA carnitine like we did outside. Okay, so that CPT2 just adds the CoA. So CPT1 takes the CoA out so we can let it inside, and then CPT2 puts the CoA back on. Okay. So what's really important here is to know that malonyl CoA, which you guys talked about in fatty acid synthesis, malonyl CoA is one of the like most important things we need to give us a fatty acid. So when we have a lot of malonyl CoA, that means we're fatty acid synthesis is working. That means we're making a lot of fatty acids. That malonyl CoA inhibits CPT1. So it makes sense because we wouldn't need when we have a lot of fatty acids or malonyl CoA. When we have a lot of that, we don't want to also break any. If we're making a lot of fatty acids, we don't want it to be happening at the same time of breaking down a lot of fatty acids. Does that make sense? Because it wouldn't make sense for us to be making fatty acids and breaking down fatty acids at the same time. So malonyl CoA inhibits CPT1, and that is really important. That is that's very high yield. Like it'll for sure come as a question. Okay. So the third step. So that's the second step. Now we're inside. So first of all, we got activated by ALCAS, and then it got transported by CPT1, translocase, CPT2. Now we have it inside the mitochondrial matrix. Now we go through the 4 different like small reactions to give us to break down that fatty acid, like ultimately. Okay. So those 4 steps is a process. That process, every time a fatty acid goes through a process, it decreases it by 2 carbons. So we go down this process. So let's say we have C16. Okay, it goes through this process, and this process, and this process, and this process. Now we have C14. Okay, it goes back up. Now we have C14. It goes through the process again until we have C12. Okay, so every process brings down the fatty acids by 2 carbons. Okay, so the four reactions are: reaction 1 is oxidation or dehydrogenation. It's the same name. Okay, so don't get confused because it's written here dehydrogenation. It's the same thing as oxidation. So it's that process is done by acyl-CoA dehydrogenase. Okay, acyl-CoA dehydrogenase, and it says family. Why does it say family? Because let's say we don't always have a C16 fatty acid, right? Because we don't always eat the same fatty food. Okay, so that means for each, like for example, C16, we will need a C16 acyl-CoA dehydrogenase specific for that fatty acid. But for C14, same thing. We need another acyl-CoA dehydrogenase. Does that make sense? Okay. So this enzyme comes in and it oxidates, which means it just adds a double bond. Okay. And here we get we get FADH. Okay. And then we get hydration. by anyl-CoA hydratase, which means it just adds water to it. And then we get another oxidation, which is by the 3-hydroxyl-acyl-CoA dehydrogenase, and that gives us an NADH. And then finally, we will, by beta-keto-acyl-CoA thiolase, we get our acetyl-CoA.
We can't hear you.
Hello?
Oh, hello.
Sorry about that. I don't know what happened. It just kicked me out of nowhere.
No, that's all right.
Okay. Where were we? So yeah, so can you just give me the so I can share my screen, the host. Thank you.
Sorry.
That's okay. All right. Okay, so we just finished. Okay, we just finished those 3 steps. Do you guys have any questions? Do you guys have any questions so far? Anything that doesn't make sense?
Can you repeat them again, please?
Which ones? The last 4 steps?
Yes, yes.
Okay, sure. Okay, so now, I just want to orient you. Right now, we're inside the mitochondrial matrix. Okay, what we have is a fatty acyl CoA. Okay, that fatty acyl CoA goes through 4 different reactions, and those reactions are here. So it's called acyl CoA dehydrogenase, which is it's a family because it has different enzymes for different fatty acids. So, for example, a C16 fatty acid needs a different enzyme than like it's probably called like a 16 acyl CoA dehydrogenase. It needs a different one than a 14 carbon fatty acid. So we use this enzyme, which is the general name of the enzyme, to put a double bond in this. So So here we just put a double bond. So here we don't have a double bond. Here we have a double bond. So this stuff you guys don't really need to know. You just need to know that it goes through those different reactions, which is oxidation, hydration, oxidation, and then cleavage. To understand what's going on, you guys don't need to know the whole structure. Okay, so that first reaction, which is an oxidation, reduces FAD to FADH2, and that FADH2 can be used in the citric acid cycle. Okay, then we have hydration, where we take the water out. Okay, the third that's by anyl cohydratase. Then we have a third reaction, which is the second oxidation, and it's done by 3-hydroxyacyl CoA dehydrogenase. And then here it's important because we get the NADH2. Again, NADH. The NADH can then be used in the citric acid cycle again. And then we'll go to acetyl CoA, where we get acetyl CoA, and it's by beta ketoacyl CoA thiolase. Okay. So now, so as I said, so here let's say we have a C16 fatty acid. Okay. Every time it goes through this whole cycle, it becomes 2 carbons shorter. Okay. So it becomes, instead of becoming C16, C being carbon, it becomes C14 fatty acid. Okay, so that keeps happening. It keeps going through that process every time, losing 2 carbons until we're done, until we have all acetyl CoA we need, and we have the NADH and FADH that we can use in the citric acid cycle. Okay, does that make sense?
Yes.
Perfect. Okay, so I skipped. I put some of the clinical slides at the end because I just like teaching like normal, what happens normally, and then what happens in like abnormal situations. So CPT1, okay? So here it's telling us that CPT1, when CPT1 is associated with type 2 diabetes and insulin resistance, okay? He's just telling you that it's related to those 2 diseases. So for example, when we have insulin resistance, so when there's a lot of insulin being produced, we are increasing glucose, okay, and we're increasing glucose, and we're increasing something that you guys learned in fatty acid synthesis, how insulin also increases fatty acid synthesis. It increases myelin CoA, and that ultimately increases fatty acids. So because it's increasing myelin CoA, that myelin CoA inhibits CPT1, as we said here. CPT inhibits myelin CoA. So when we have a lot of insulin, that insulin will act in the fatty acid synthesis pathway and activate it more than it's already active, and it will yield a lot of myelin CoA. That myelin CoA will go back and inhibit CPT1. So that means we'll have decreased oxidation of long chain fatty acids. That means we'll have a lot of fatty acids. We'll have a lot of long chain fatty acids, and we'll have an increase in free fatty acids as well. Okay. We just have a lot of free fatty acids just bound to albumin. Okay, we'll have a lot of free fatty acids in the bloodstream. Okay, and then he talks here about the carnitine deficiency. So it's the carnitine deficiency. It's a genetic disorder, and it comes by age 5. Symptoms like hypoglycemia and skeletal muscle weakness, cardiomyopathy should be memorized. You guys should just know that like this is what happens with carnitine deficiency. And why does it happen? Again, as we said up there. So when there is no carnitine, there is no fatty acid breakdown. So that means we're not getting enough ATP. We're not getting enough acetyl CoA. That means we're not getting enough ATP, which can cause the skeletal muscle weakness. So like, because our muscles like mostly use fatty acids for ATP. So that means when we're not breaking down fatty acids, we're not getting enough ATP. Okay, this is what happens usually in a carnitine deficiency, like physiologically. Okay, here he's talking about another deficiency that comes in a myopathic form. So he's just talking about it's CP2 deficiency, which again, CP2 we said is going to be the enzyme. I'm going to go up to the lecture. Yeah, so it's going to be the enzyme that gives us acyl-CoA plus carnitine from acyl-carnitine. So it just adds the CoA, just adds that CoA inside the matrix. Okay, that's what it does. So when we have a deficiency in that enzyme, it usually comes in a myopathic form. Do you guys know what myopathic means? Myopathic just means it's related to the muscle. So we get a lot of muscle aches. We get myoglobinuria, so red urine. We get those, and then those muscle aches and stuff can exacerbate, can get worse when there's prolonged exercise or when there's a lot of fasting or stress. Okay. And then so the primary treatment, you guys, we won't talk about treatment a lot in MOL 2, but like it's to basically just stop doing activities, because the more you do activities, the more we need ATP. But again, there's no carnitine, that means there's no ATP, there's no acyl-CoA, so we won't have enough energy to do the exercise, and we don't have enough energy to fast because we can't break down those fatty acids into energy. Okay. A medium chain acyl-CoA dehydrogenase. This is high yield as well. This will be a question on the exam for sure. It's really high yield. Even USMLEs, I hear a lot about it. So it just means, so we know how we talked about the acyl-CoA dehydrogenase family. We like here, acyl-CoA dehydrogenase family. Yeah, so here we're talking about one that is specifically for medium chain fatty acids. So this one is common. The deficiency is common. It happens from C8 to C10. Okay, so it's an enzyme that is supposed to be breaking down C8, C9, and C10 fatty acids. So when it doesn't break them down, that means we'll have them a lot in our blood. And we have them a lot in our blood. So the patients will present with hypoglycemia. They will present with even with fasting, they'll get hypoglycemia because there's lack of ATP. So when we're fasting, we're not getting enough ATP to fuel the gluconeogenesis, because in gluconeogenesis we need some ATP. So we don't have ATP to fuel gluconeogenesis, hence we get hypoglycemia. Okay, and again, so like we don't get fatty acids, so we don't get acyl-CoA. Then acyl-CoA cannot make ATP. We don't have ATP for gluconeogenesis, hence we just have hypoglycemia. Okay. So we'll have low to absent ketones. Low to absent ketones. Why? Because we're not breaking down that fatty acid, we're not getting acyl-CoA, and we need acyl-CoA to make the ketones. Okay, we'll get to know in the last two slides here. Okay, so peroxisomes, this is just this slide has nothing to do with like the lecture. It's just I think the doctor put it there because this is something that we need to know. So peroxisome, so that whole like fatty acid oxidation happens in the peroxisome, but in the peroxisome something else happens, is it has a catalase enzyme, and that catalase enzyme breaks down H2O2, which is hydrogen peroxide, okay, into water and molecular oxygen. So acyl-CoA dehydrogenase, which is again the same one we talked about right here. So this one in the peroxisome, it transfers electrons to O2. So it basically gives us hydrogen peroxide, which is toxic. So in the peroxisome, the acyl-CoA dehydrogenase doesn't do the normal thing it does in the mitochondria. It gives you hydrogen peroxidase, and then peroxide. It gives you hydrogen peroxide, and then that hydrogen peroxide by the enzyme catalase gives you water and oxygen. Okay, it's just stuff that you guys need to know, but it's nothing. It's not important. It's not that important for the understanding of this lecture. Okay, now so that whole lecture, those whole slides were talking about even number fatty acids. So in odd number fatty acids, so the odd chain fatty acids, there's different things that happen. Okay, so let's say we have, this is a C3. So you see 1, 2, 3. So this is a C3. When we have an odd number fatty acid, we use two different enzymes. We'll use, and those two are also very, very important, okay? Those two are called propionyl CoA and methyl malonyl CoA. Those two give you succinyl CoA, which again go into the citric acid cycle and do the same job, or almost the same job that acetyl CoA did, okay? So it just uses different enzymes. The propionyl CoA, carboxylase, this is the enzyme. It's important because it needs a coenzyme. That coenzyme is biotin, okay? And from methyl, methyl malonyl CoA, we have an enzyme called methyl malonyl CoA mutase, which is a cofactor called B12. Okay, so this stuff you just have to remember, but it's basically it's really high yield, and I think you guys probably need to know that like biotin, it's in propionyl CoA carboxylase is biotin, or the same thing, it's called B7. Okay, and in this enzyme methyl malonyl mutase, you need B12. It's the same thing. It's just another name for it, or cobalamin. Okay, so it's important to know that biotin is the same thing as B7, and B12 is the same thing as cobalamin. Because maybe in like the exam they give you like B7, B3, B6 like that. So you guys need to know like in case they give you the number or the actual name of it. Okay. Okay, so now we're going to talk about ketone bodies. Okay, so that acetyl CoA that we broke down from fatty acids. Yeah, so that acetyl CoA that we broke from fatty acids, it needs oxaloacetate. In the citric acid cycle, if you guys remember, we need oxaloacetate for the acetyl CoA to work. If there is no oxaloacetate, if oxaloacetate is missing, is limiting, then acetyl CoA, instead of going into the TCA cycle, it forms ketone bodies. How does it form ketone bodies? It forms, so those enzymes, like these enzymes and those names, I don't think you guys need to know. Maybe this one you guys need to know, but the one that you need to know for sure is this one, because it's the rate limiting step. And you guys probably know what rate limiting step. It just means it's the enzyme, it's the step that usually gets regulated. Okay, so the first step is we have the acetyl CoA. Then we get an acetyl CoA, and then with the help of HMG CoA synthase, we get HMG CoA, and then acetoacetate. The acetoacetate is the ketone body we use. Okay, and yeah, so acetoacetate gives you this guy, the 3-hydroxybutyrate and acetone. Here, I remember when I was studying this lecture last year, it's something like made me remember like very well what ketone bodies is when there's, have you guys heard of diabetic ketoacidosis? So it's basically when, so diabetes, when a person with diabetes, it usually messes up the TCA cycle. When it messes up the TCA cycle, because it messes up because this person has diabetes, it messes up the hormones, so they're insulin and glucagon, and therefore that acetyl CoA cannot be used in the TCA cycle and ends up being used in ketone bodies, in ketone body formation. So because diabetic ketoacidosis is known to have patients come in with acetone smelling breath. Okay, So this is how this I kind of like remember that as a story kind of thing, and it made me remember that when acetyl CoA cannot be used in the TCA cycle because of oxaloacetate, then it gets directed into making ketone bodies. Okay. Now, there's an important thing to know about ketone bodies is that so they are made in the liver. Okay, but there is something cool about them is that not a lot of organs in our body can use those ketone bodies, except for the muscles, and we have the brain. So we didn't talk about the brain in this lecture, but he did talk about the muscles. So you see this 3-hydroxybutyrate. So that was the end result here of what's it called, of ketone bodies. So that thing can go into the muscles, and because the muscles have this enzyme thiophorase, we can actually utilize the ketone body into making acetyl CoA. Okay, so we can degrade that ketone body again into an acetyl CoA, which then can be used for us to get more ATP. Okay, so it's important to know here that in this lecture we only talked about the muscles having the enzyme that can yield the acetyl CoA. Okay, and that's all I think he wanted you guys to know in this slide. Do you guys have any questions so far? We're almost done. We just have a bunch of questions I just prepared for you guys.
Can you briefly re-explain the ketone bodies and how it got formed?
How they get formed?
The last slide about
Last slide.
acetoacetate and
Okay. Yeah. So here what he's highlighting here. So like don't look at all those enzymes that are written and stuff. Those are not like high yield. What he wants you guys to know from this slide is that ketone bodies are made from acetyl CoA. When acetyl CoA cannot go into, so if acetyl CoA cannot go through TCA cycle because there's low levels of oxaloacetate, that acetyl CoA is then utilized to make ketone bodies. And by ketone bodies, we mean those acetoacetate or the 3-hydroxybutyrate. That happens in the liver, okay? So it happens in the liver, and we cannot utilize it. Cannot utilize it to make energy, right? But in the muscles, because it has the enzyme called thiophorase, that enzyme makes us able to transfer this ketone body, those ketone bodies, makes us transfer those ketone bodies into 2-acetyl CoA, okay? And those acetyl CoA can be used then in the muscles. That's all he wanted you guys to know from this slide. There's nothing else that he's highlighting here.
Okay, thank you so much.
Of course. I'm just gonna go over some questions, just three or four questions. If you can just write what you think the answer is. So a parent of a 3-month-old. So first of all, I'm just gonna go over how I would solve this question. So the way I like to solve questions like this is because when you have a MOL exam, you have around 80 to 100 questions, and they're all kind of clinical scenarios. So what I like to do is I like to look at the answers first and kind of see where what lecture I should be looking at, or what's something I need to remember. So here I would look at the options, and then I would read the question. Okay, so here we have a 3-month-old infant who is extremely lethargic, and those examinations show someone who's hypoglycemic. So that means they have low blood glucose and low measurable ketones. So basically they have no ketones in the urine and blood. Okay, the blood indicates an elevation in butyric acid, whatever, and it shows in the blood analysis has C8-acylcarnitine. A deficiency of which of the following is responsible for this observation? What do you guys think? Okay, so I see some right answers. Yes, so it is E. So from this whole, what you guys need to know from this whole lecture, from this whole question, it's basically telling you there's a person that has C8, like acylcarnitines, so C8 fatty acids in their blood, and that they have low measurable ketones. So they have C8 and they have low ketones. That means this person cannot break down those C8 fatty acids, and therefore cannot, and they don't have ketones. Therefore, it has to be MCAD, or medium chain acyl CoA, because again, it's C8 to C10. Okay, so I have some explanations here. I'm gonna send this lecture to one of your BLCs, and they can share it if you guys would like to look at it again. So this is the second question. What do you guys think? So oxidation of fatty acid requires the input of energy in the form of ATP. Which of the following activities requires this energy? So remember, we talked about an enzyme that requires 2 ATPs. Which one is it? What do you guys think? Okay, well, this is just a straight up recall question. So the only enzyme that needs ATP and fatty acid oxidation is the acyl CoA synthetase. Okay, you can see the explanation here. Okay, third question. An individual suffers from a defect in one of the enzymes required for synthesis of carnitine. If the individual does not have adequate intake of carnitine in the diet and is fasting, which of the following will be most likely observation in this person compared to conditions of normal carnitine? So basically, this whole question, this whole thing is asking you about the function of carnitine, and the function of carnitine, what it does, and what would it do, what would happen if it wasn't there. What do you guys think the answer is? What do you guys think? Okay, so yeah, so it will be elevated long chain fatty acids, because so when we don't have carnitine, that means we have a lot of fatty acids that cannot enter the mitochondria for the beta oxidation. So that just means we have elevated long chain fatty acids. Okay, last question. When fatty acids with odd numbers of carbon atoms are oxidized in the beta oxidation pathway for the final product, which is acetyl CoA and 1 mole of the 3 carbon molecule, propionyl CoA. In order to use the propionyl carbons, the molecule is carboxylated and converted ultimately to succinyl CoA and fed into the TCA cycle. Which of the following represents the vitamin cofactor? So we're talking about a cofactor that is needed in the propionyl CoA pathway. Remember, we talked about there's 2 enzymes and there's 2 cofactors that we need. So which of these 4 you guys think?
B12.
Yes, so B12. So this one is as B12, because we talked here about B12. Yes, B12 and B7. Okay. Yeah, and that will be all for us, guys. If you see me, if you have any questions, let me know. I have my phone number and I have my email up here. So if you guys have any questions, let me know, and I will be happy to help. Thank you for listening today, and Allah waffaqkum, inshallah. Good luck.
Thank you so much. It was very
Of
helpful.
course.